Welcome, everyone. I'm excited to kick off our presentation today with the fascinating topic, fundamental unit of life. On this first slide, you'll see the title and subtitle, which set the stage for what we'll explore. Over the next few slides, we'll dive into what makes cells the building blocks of all living things, their structure, and their importance. So let's get started and uncover the mysteries of the fundamental unit of life. Let's take a look at the contents of this presentation. We start with the discovery of the cell, which is a fundamental milestone in biology. Understanding organisms is next, and that includes both unicellular and multicellular forms. Moving forward, we'll explore different types of cells in the human body and their characteristics, which help us appreciate the diversity and complexity of life. We'll then examine the structure of a typical cell, including key components like the cell membrane or plasma membrane, which controls what enters and exits the cell. From there, we'll discuss cell wall, which provides additional support in plant cells, and delve into important cell organelles such as the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. Understanding how substances move in and out of cells is crucial, so we'll cover concepts like osmosis and diffusion, along with solutions like isotonic, hypotonic, and hypertonic, which affect cell stability. We'll also compare animal and plant cells to highlight their similarities and differences. Finally, we'll look at specialized structures called plastids in plant cells and the overall comparison between animal and plant cells, tying everything back to the central theme of understanding cell structure and function as the foundation of biology. So let's start with the discovery of the cell itself. Cells were first identified by a scientist named Robert Hooke back in 1665. He was experimenting with a microscope he designed himself, which was quite an achievement for that time. When he looked at a thin slice of cork, yes, cork from the bark of a tree, he noticed that it had a very peculiar structure. It looked like a honeycomb with many small, box-like compartments. These tiny chambers reminded him of the cells in a monastery, which is how they got the name. He called these small units cells, and that marked the beginning of cell biology. This discovery was fundamental because it opened the door to understanding the basic building blocks of all living organisms. It's fascinating to think that this simple observation led to such a huge scientific field that helps us understand life today. Let's take a closer look at Robert Hooke's microscope. This device was a groundbreaking instrument in the history of science. Hooke's microscope was one of the earliest microscopes capable of magnifying objects enough to reveal details invisible to the naked eye. It played a crucial role in advancing our understanding of the microscopic world. With this microscope, Hooke was able to observe and describe the tiny structures in a slice of cork, which he famously called cells. This discovery laid the foundation for cell theory, a fundamental concept in biology. Overall, Hooke's microscope exemplifies how improving tools can dramatically expand our ability to explore and understand nature. So, Robert observed that cork actually comes from the bark of the tree, which is quite fascinating because it's a natural material that's harvested without harming the tree. This process allows the tree to continue growing while providing us with cork, which has many uses. And then there's honeycomb. Now, honeycomb is that amazing structure made by bees, and it's a perfect example of nature's engineering, strong, lightweight, and efficient. Both cork and honeycomb highlight how natural materials are designed by nature to serve specific functions, and they remind us of the incredible resourcefulness found in the natural world. These observations tie into our overall theme of exploring sustainable and innovative materials inspired by nature. Let's talk about organisms and how they can be classified based on their cellular structure. First, we have multicellular organisms, which are made up of many cells. Think of humans, elephants, cows, and even plants like oaks and pines. These organisms have specialized cells that work together to keep everything running smoothly. On the other hand, there are unicellular organisms, which consist of just a single cell. Examples include amoebas, paramecia, and chlamydomonas. Despite being just one cell, these organisms are incredibly capable and can perform all necessary life functions within that single cell. Understanding the difference between multicellular and unicellular organisms helps us see how life is organized and how complexity arises. It's a fundamental concept that ties into the bigger picture of biological diversity and how organisms survive and adapt in their environments. All right, on this slide, we're focusing on unicellular organisms, which are fascinating because they are made up of just a single cell. Two great examples to keep in mind are Chlamydomonas and Paramecium. Chlamydomonas is a kind of green algae, and it's often studied for its simple structure and how it moves using tiny flagella. Paramecium, on the other hand, is a ciliate protozoan that moves with hair-like structures called cilia, helping it navigate its environment. These organisms are perfect examples of how complex life functions can be carried out by just one cell. They also remind us of the incredible diversity and adaptability of single-celled life forms. This ties into our overall theme of understanding the building blocks of life and how even the simplest organisms have sophisticated mechanisms for survival. Now, let's move on to the next category of multicellular organisms. When we talk about multicellular organisms, we're referring to living things made up of many cells working together. These organisms are incredibly diverse and include animals, fungi, and plants. Animals are perhaps the most familiar. They have specialized cells that perform specific functions, allowing them to move, respond to their environment, and carry out complex behaviors. Fungi are another group. They include mushrooms, yeasts, and molds. Fungi play essential roles in ecosystems, especially in decomposing organic material and recycling nutrients. Then we have plants, which are vital for life on Earth. They use photosynthesis to produce their own food and provide oxygen and food for many other organisms. Connecting this to our overall theme, understanding these groups helps us see how different multicellular life forms have evolved unique structures and functions to survive in their environments. Each group plays a crucial role in maintaining the balance of life on our planet. Let's take a closer look at the different types of cells in the human body, starting with the nerve cell, which is essential for transmitting signals throughout our body, allowing us to think, feel, and move. Moving on, we have blood cells, which play a vital role in transporting oxygen and nutrients, as well as fighting off infections. Next is the fat cell, or adipocyte, responsible for storing energy in the form of fat. Then there's the sperm cell, a specialized reproductive cell that carries genetic information for reproduction. Following that, we have the bone cell, which helps maintain and strengthen our skeletal structure. Lastly, the ovum, or egg cell, is crucial for reproduction as the female reproductive cell. Each of these cells has a unique structure and function, but together, they highlight the incredible diversity and specialization within our bodies. This diversity is what enables us to perform a wide range of biological functions seamlessly. Now, let's talk about the cell. A cell is actually the smallest unit that can perform all the essential life functions. Think of it as the basic building block of life. Everything living, from tiny bacteria to complex humans, is made up of cells. They're incredibly versatile and capable of carrying out processes like growth, energy production, and even reproduction. Understanding cells is key because they form the foundation for how all living organisms function and survive. So, when we discuss the structure and function of organisms, we're really talking about these tiny but mighty units, the cells. Let's talk about cell characteristics. The shape and size of cells are actually closely related to the specific functions they perform. For example, some cells are long and thin, while others are more rounded, all depending on what they need to do. Every living cell has the basic ability to carry out certain essential functions that keep it alive and working. Inside each cell, you'll find a variety of components called organelles. Think of organelles like tiny specialized parts, each with its own unique job. For instance, some organelles are involved in energy production, while others handle protein synthesis. What's interesting is that all cells, regardless of their type or the organism they belong to, have the same set of these organelles. This commonality highlights a fundamental aspect of biology. Despite the incredible diversity of life, there are shared cellular features that form the foundation of all living organisms. Connecting this to the overall theme, Understanding these cell characteristics helps us appreciate how form and function are so intricately linked at the cellular level. Now, let's take a closer look at the cell membrane, also known as the plasma membrane. This is the outermost layer of the cell, acting as a protective barrier that separates the interior of the cell from the external environment. It plays a crucial role in controlling what enters and exits the cell, ensuring that essential nutrients get in and waste products are removed. One of the most interesting features of the membrane is its ability to be selectively permeable, meaning it allows some materials to pass through while blocking others. This selectivity is vital for maintaining the cell's internal balance or homeostasis. The plasma membrane is also quite flexible, which helps the cell adapt and move as needed. It's primarily made up of organic molecules like proteins and lipids, which work together to give the membrane its structure and functions. Understanding this membrane helps us see how cells interact with their environment, which is a key part of understanding cellular function as a whole. Now, let's take a look at how materials move through the cell membrane. There are two main types of movement that we need to understand. The first is gaseous exchange, which primarily happens through diffusion. This is when gases like oxygen and carbon dioxide pass directly through the membrane, moving from areas of higher concentration to lower concentration without the need for energy. The second type is liquid exchange, which occurs through osmosis. Osmosis involves the movement of water molecules across the membrane, again from an area of lower solute concentration to an area of higher solute concentration. Both of these processes are essential for maintaining the cell's internal environment and ensuring it functions properly. They connect directly to our overall theme of how cells interact with their surroundings and regulate what enters and leaves. Understanding these movements helps us see how vital the cell membrane is in controlling the cell's internal conditions. Now, let's talk about the movements of materials through the cell membrane, starting with osmosis. Osmosis is essentially the movement of water from an area where there's a high concentration of water to an area where there's a lower concentration. This movement happens through a semi-permeable membrane, which allows water to pass, but blocks many other substances. It's a crucial process because it helps maintain cell balance and hydration. Additionally, gaseous exchange, like oxygen and carbon dioxide, also moves from higher concentration areas to lower concentration areas, following the same principle. Understanding these processes is key because they illustrate how cells regulate their internal environment and interact with their surroundings. This movement of water and gases is fundamental to many physiological functions and overall cellular health. Let's take a closer look at the difference between osmosis and diffusion. Diffusion is a process that can happen in any kind of medium, solid, liquid, or gas. It doesn't need a special barrier. It just occurs naturally as particles move from an area of higher concentration to an area of lower concentration. On the other hand, osmosis is a specific type of diffusion that happens only in liquids. It involves the movement of water molecules through a semi-permeable membrane, which allows water to pass but blocks certain solutes. So, the key points to remember are that diffusion is more general and can occur in any medium without special structures, while osmosis is exclusive to liquids and requires that semi-permeable membrane to control the movement. Understanding this distinction helps us see how these processes are fundamental to biological functions and how substances move within organisms and their environments. Today, we're going to look at 3 solutions that are key to understanding how substances move across cell membranes. First, we have the hypertonic solution. This is when the solution outside the cell has a higher concentration of solutes compared to the inside. As a result, water tends to move out of the cell, causing it to shrink. Next, there's the isotonic solution. In this case, the concentration of solutes outside and inside the cell are equal. This balance means water moves in and out at the same rate, keeping the cells stable. Finally, we have the hypotonic solution. Here, the outside solution has a lower concentration of solutes than the inside of the cell. Water moves into the cell, which can cause it to swell or even burst if the influx is too much. Understanding these solutions helps us see how cells regulate their internal environment and respond to different conditions. Now, let's talk about isotonic solutions. When the medium surrounding a cell has the same concentration of water as the inside of the cell, we call that an isotonic environment. In this case, water molecules move across the cell membrane in both directions, going in and out. But the key point is that the amount moving in equals the amount moving out. So, there's no overall movement of water, and the cell maintains its size. This balance is really important because it helps cells stay stable and function properly within their environment. Understanding isotonic solutions is fundamental to grasping how cells interact with their surroundings, which ties into the broader theme of how cells regulate their internal and external environments to stay healthy. Now, let's talk about hypotonic solutions. A hypotonic solution is essentially the fluid surrounding the cell that has a higher concentration of water compared to the inside of the cell. Think of it as the environment being more water-rich. Water molecules can move across the cell membrane in both directions, but because there's more water outside, more water tends to flow into the cell than out. This net movement causes water to enter the cell, and as a result, the cell is likely to swell up. This concept is important because it highlights how the balance of water and solutes influences cell size and function, which ties into the broader theme of how cells respond to their environment. Now, let's talk about hypertonic solutions. When the environment surrounding a cell has a lower concentration of water compared to what's inside the cell, we call that a hypertonic situation. In this case, water still moves across the cell membrane in both directions, but more water tends to leave the cell than enters. As a result, the cell begins to shrink or shrivel. This concept is important because it highlights how the balance of water and solutes between the cell and its environment can affect cell size and function. Understanding hypertonic solutions helps us grasp how cells respond to different environments, which is fundamental to many biological processes. Now, let's talk about plasmolysis. This is a fascinating phenomenon that happens in plant cells. Essentially, when a plant cell loses water through osmosis, the cell membrane pulls away from the cell wall. This shrinking process is what we call plasmolysis. It occurs because the water inside the cell decreases, causing the cell to lose turgor pressure. The most important point to remember is that plasmolysis indicates a loss of water, often due to hypertonic environments. This process highlights how vital water balance is for maintaining cell structure and function. Connecting this to the overall theme, understanding plasmolysis helps us see how cells respond dynamically to their environment, especially when water availability changes. Let's take a closer look at the cell wall. It's the hard outer covering of the cell, positioned outside the cell membrane. Think of it as a tough armor that protects the cell from external forces. The main component of the cell wall in plant cells is cellulose, which is a complex carbohydrate that gives the wall its strength. This structural support is crucial because it helps the plant maintain its shape and withstand environmental changes like wind, rain, or even mechanical stress. So, in the broader context of cell structure, the cell wall plays a vital role in ensuring the plant's stability and survival in its environment. Let's take a closer look at the nucleus, which is often called the control center of the cell. It has a double-layered covering known as the nuclear membrane, acting like a protective barrier. This membrane isn't just a simple boundary, it has tiny pores, which are crucial because they allow materials like molecules and signals to pass in and out of the nucleus. This exchange is vital for the cell's functions. Inside the nucleus, you'll find chromosomes. These are the structures that only become visible as rod-shaped entities when the cell is about to divide. Think of chromosomes as the cell's instruction manuals. They carry the genetic information needed to pass features from parents to offspring. This information is stored in the form of DNA molecules, or deoxyribonucleic acid, which contains the blueprint for all the traits and functions of living organisms. Understanding how the nucleus manages genetic information helps us see the bigger picture of how cells grow, divide, and pass on life's instructions. Now, let's talk about the cytoplasm. This is the fluid content that fills the inside of the cell, essentially the space inside the plasma membrane. It's not just empty space, it's a dynamic environment that supports the cell's activities. Within the cytoplasm, you'll find various specialized cell organelles, each with its own unique function. These organelles work together to keep the cell alive and functioning properly. Understanding the cytoplasm helps us see how the cell maintains its structure, transports materials, and carries out vital processes, all crucial for the overall health and function of the organism. Let's take a closer look at some essential cell organelles that keep the cell functioning smoothly. First up is the endoplasmic reticulum, often called the ER. It acts like a manufacturing and packaging system, synthesizing proteins and lipids and transporting them where needed. Next, we have lysosomes, which are like the cell's cleanup crew. They break down waste materials and cellular debris, maintaining a healthy environment inside the cell. Moving on, the Golgi apparatus functions as the cell's post office. It modifies, sorts, and ships proteins and lipids to their final destinations, ensuring everything gets where it should go. Then we come to plastids, which are primarily found in plant cells. They're involved in storing pigments and nutrients, and some, like chloroplasts, are vital for photosynthesis, the process that allows plants to convert sunlight into energy. Speaking of energy, mitochondria are often called the powerhouse of the cell. They generate most of the cell's energy through respiration, powering all cellular activities. Lastly, vacuoles. These are like storage tanks. They hold water, nutrients, or waste products, and in plant cells, a large central vacuole provides structural support and maintains turgor pressure. By understanding these organelles, we see how each part plays a crucial role in maintaining the overall health and functionality of the cell, which ties back to our theme of cellular complexity and efficiency. Let's take a closer look at the endoplasmic reticulum, or ER for short. The ER is a large, complex network made up of membrane-bound tubes and sheets that extend throughout the cytoplasm. It's really like a busy highway system within the cell, facilitating the transport of materials. There are two main types of ER, the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or SER. The RER has ribosomes attached to its surface, giving it a rough appearance, and it's primarily involved in protein synthesis. On the other hand, the SER lacks ribosomes and is more involved in lipid production and detoxification processes. Both types of ER serve as channels, helping to move materials efficiently between different regions of the cytoplasm, or even between the cytoplasm and the nucleus. Think of it as a kind of cellular highway that keeps everything moving smoothly. Additionally, the ER provides a sort of cytoplasmic framework, sort of like scaffolding, that supports various biochemical activities. This structure is essential for maintaining the cell's organization and ensuring that everything functions harmoniously. So, in summary, the ER is crucial for transport, structural support, and biochemical processes, making it a central player in maintaining cellular health and activity. Let's take a closer look at the Golgi apparatus, which was first discovered by Camillo Golgi. It's essentially a series of membrane-bound vesicles that are stacked parallel to each other, and these stacks are called cisterns. Think of it as a processing and sorting center within the cell. The Golgi is responsible for handling materials that are synthesized near the endoplasmic reticulum, or ER. It packages these materials into vesicles and then dispatches them to various destinations, both inside and outside the cell. This makes it a crucial part of cellular logistics and communication. Its main functions include storage, modification, and packaging of proteins and other products. These are then sent to their correct locations. Additionally, the Golgi plays a key role in forming lysosomes, which are the cell's waste disposal units. Overall, the Golgi apparatus is vital for ensuring that proteins and other molecules are properly processed and directed, supporting the cell's overall function and health. Now, let's talk about lysosomes, which are essentially the waste disposal system of the cell. These tiny structures are responsible for keeping the cell clean by digesting any foreign material that enters, as well as worn-out or damaged cell organelles. They contain powerful digestive enzymes, which are capable of breaking down all kinds of organic material. This means they play a crucial role in recycling and maintaining cellular health. Interestingly, when a cell gets damaged or is under stress, lysosomes can sometimes break open and release their enzymes inside the cell itself. This process leads to the digestion of the cell, which is why lysosomes are sometimes called the suicide bags of the cell. It's a fascinating balance. These structures are vital for cleaning up and recycling, but they can also lead to cell death if things go wrong. This highlights how important lysosomes are in overall cell health and maintenance. Let's talk about mitochondria, often called the powerhouse of the cell. These tiny structures are essential because they produce the energy our cells need to carry out a variety of chemical activities. They do this by converting nutrients into ATP, which stands for adenosine triphosphate. Think of ATP as the energy currency of the cell. It's what powers everything from muscle contractions to nerve signals. Mitochondria have a unique structure with two membranes. The outer membrane is quite porous, allowing molecules to pass in and out easily. The inner membrane, on the other hand, is deeply folded into structures called cristae. This folding increases the surface area, which is crucial for energy production. Interestingly, mitochondria are semi-autonomous. They can produce some of their own proteins because they have their own DNA and ribosomes. This self-sufficiency allows them to carry out specific functions independently from the rest of the cell. Connecting this to our overall theme, understanding mitochondria highlights how cells are energy factories, specialized, efficient, and capable of maintaining their own functions to keep us alive and active. Now, let's talk about plastids, which are fascinating organelles found in plant cells, as well as in algae and some other eukaryotic organisms. These are membrane-bound structures that play crucial roles in cell function and metabolism. In plant cells specifically, we typically see two main types of plastids. First, there are chromoplasts, which are responsible for giving color to fruits and flowers by storing pigments. These are the colorful plastids you often see. Then we have leucoplasts, which are generally colorless and mainly involved in storing materials like starch, oils, and proteins. A key point to remember is that plastids containing green pigment, which is chlorophyll, are called chloroplasts. These are essential for photosynthesis, allowing plants to convert light energy into chemical energy. Now, despite their differences, all plastids share some common features. They contain membrane layers embedded in a fluid called stroma, which is rich in enzymes and other molecules. Structurally, plastids resemble mitochondria. They even have their own DNA and ribosomes, which means they can produce some of their own proteins independently. Understanding plastids helps us appreciate how plants carry out vital processes like photosynthesis and storage, which are fundamental to life on Earth and directly connect to the larger theme of how plant cells sustain themselves and support ecosystems. Vacuoles are essentially storage sacs within cells, and they can hold either solid or liquid materials. In animals, these vacuoles tend to be smaller, while in plant cells, you'll find large vacuoles that are quite prominent. These large vacuoles are filled with cell sap, which helps give the plant cell its turgidity and rigidity, so they stay firm and upright. They also serve as storage units for various substances like amino acids, sugars, organic acids, and some proteins. For example, in amoeba, the food vacuole is where the organism stores the food it has just consumed. Another key role of vacuoles is helping the cell manage water and waste. They expel excess water and some wastes, which is crucial for maintaining the cell's internal balance. So overall, vacuoles are vital for storage, structural support, and waste removal, all of which are essential for the cell's health and functionality, tying into the bigger picture of cell organization and survival. Let's take a look at how animal and plant cells compare. Generally, animal cells tend to be smaller in size, making them more compact, while plant cells are usually larger, which supports their need for storing more materials. One key difference is that animal cells lack a cell wall, unlike plant cells that have a rigid cell wall providing structure and support. When it comes to plastids, most animal cells don't have them at all, except for the protozoan euglena, which is a bit of an exception. Plastids, like chromoplasts and leucoplasts, are present in plant cells and are vital for functions like pigment storage and carbohydrate synthesis. Vacuoles are another important point. Animal cells do have vacuoles, but they tend to be smaller, whereas plant cells have larger vacuoles that help maintain cell turgor and store nutrients. So, understanding these differences really highlights how each cell type is specialized to fulfill its role. Animal cells for mobility and versatility, and plant cells for structure and energy storage. Thank you for your attention.